For decades, biology operated on a strict binary assumption. All life on Earth was divided into two distinct categories: simple prokaryotes, like bacteria, and complex eukaryotes, encompassing plants and animals. Through a standard microscope, this textbook model held up. Single-celled organisms appeared effectively identical in their shape, size, and minimal internal structure. That classification was challenged in the late 1970s when biophysicist Carl Woese introduced a different metric, comparing ribosomal RNA sequences across species. This genetic mapping shattered the monolithic prokaryote group, forcing a three-part division of life: bacteria, archaea, and eukarya. While archaea mirror standard bacteria physically, their structural and genetic makeup belongs to an ancient, entirely distinct lineage. Physical shape is an unreliable metric for determining an organism's history. The true record of evolutionary lineage is encoded deep in molecular architecture. To understand the distance between these groups, we will contrast the molecular foundations of standard bacteria against the specialized structures of archaea. This comparison reveals the specific mechanisms archaea use to thrive in environments that would instantly destroy a normal cell. We will then return to the bacterial domain to examine actinobacteria, a group that ignores the typical rules of single-celled life by mimicking the complex structures of fungi. In both cases, surviving extreme physics or fierce competition requires the fundamental re-engineering of biological design. A cell membrane is an organism's primary defense. Standard bacteria construct this barrier using fatty acids and glycerol. The archaeal membrane is built differently. Instead of standard fatty acids, it utilizes branched hydrocarbon chains. The critical distinction is chemical. While bacterial membranes rely on ester bonds, archaea utilize highly resilient ether bonds. This bond chemistry provides a clear way to distinguish the two domains. Ether equals archaea. This chemistry allows archaeal tetraether lipids to form a stable monolayer, essentially fusing the typical bilayer into a single, rigid barrier. This ether-linked structure grants archaea immunity to high heat, extreme pH, and severe chemical stress. The divergence extends to the cell wall. Bacteria rely on true peptidoglycan, composed of muramic acid and D-amino acids. Archaea lack this substance entirely. Instead, archaea deploy rigid proteinaceous S layers, or a substance called pseudomurine. Pseudomurine's unique glycosidic linkages make archaeal walls fundamentally resistant to lysozyme, an enzyme that normally dissolves bacterial cells on contact. This ether-linked pseudomurine armor is the specific biochemical requirement to survive conditions that would dissolve standard bacteria. While their external armor is built for extremes, the internal machinery of archaea reveals a surprising evolutionary placement. Archaea possess complex molecular features that mirror eukaryotic biology, such as histone proteins for DNA spooling and multiple origins of replication. Even their transcription and translation machinery directly resembles that of eukaryotes, rather than bacteria. Protein synthesis follows this same eukaryotic pattern. Archaea initiate the process with methionine, rejecting the bacterial standard of N-formyl methionine. Although archaea occupy the physical shell of a simple bacterium, their genetic engine confirms they are our distant, ancient evolutionary cousins. Tools like 16S rRNA sequencing and metagenomics have mapped out a massive expansion in archaeal taxonomy. One major lineage, Euryarchaeota, houses a wide variety of specialized extremophile groups. The methanogens are a primary example. They are strict anaerobes, thriving in oxygen-free environments like wetlands and deep sediments. Their metabolism involves converting carbon dioxide, hydrogen, formate, or acetate into methane gas, driving a key portion of the global carbon cycle. In contrast, extreme halophiles inhabit high salinity biomes. They require these high salt concentrations just to maintain their cellular integrity. Species like Halobacterium utilize bacteriorhodopsin to act as a light-driven proton pump. This mechanism allows the organism to generate ATP directly from sunlight without the use of chlorophyll. Finally, the thermoacidophiles thrive in boiling, acidic environments, such as deep-sea hydrothermal vents. These organisms tolerate temperatures exceeding 100 degrees Celsius and corrosive pH levels between 1 and 2. These archaea thrive in conditions that would destroy most life. Their molecular design is optimized for the harsh extremes of early Earth. Within the bacterial domain, one group stands apart. It survives competitive environments through structural complexity rather than biochemical resistance to heat or acid. Actinobacteria are gram-positive bacteria identified by an exceptionally high G+C DNA content, typically between 55 and 75%. Despite being bacteria, actinomycetes grow in extensive branching filaments that mimic fungal networks. They utilize a biphasic life cycle, growing a substrate mycelium into a food source and an aerial mycelium upward into the air. This sequence begins with a single spore that germinates into vegetative growth, establishing the base substrate network. When the soil resources are depleted, the organism pushes vertical shoots, called aerial hyphae, upward away from the nutrient-poor zone. The tips of these aerial hyphae then fragment into long chains of spores, ready for dispersal to new environments. As the aerial mycelium forms these spores, it synthesizes secondary metabolites. Humans harvest these chemical compounds for use as clinical antibiotics. While soil-dwelling streptomyces produce medicine, related groups like Propionibacterinae are essential for cheese manufacturing and are the primary agents behind acne. Actinobacteria bypass the typical limitations of single cells by adopting fungal architecture, dominating complex microbiomes through physical expansion and chemical warfare. Viewing all single-celled life as a generic prokaryotic group ignores their actual biological sophistication. These two survival strategies illustrate a fundamental split in biological engineering. Archaea re-engineered their biochemistry to survive hostile physics and chemistry. Actinobacteria retained bacterial biochemistry, but re-engineered their physical structure into mycelial networks to overcome biological competition. This molecular diversity forced science to redraw the tree of life, positioning Archaea closer to humans than to standard bacteria. From extracting energy from light via bacteriorhodopsin to producing antibiotics to poison soil competitors, life at the microscopic level is a highly advanced engineering race. Life survives by rewriting its own operational rules, enabling it to inhabit the uninhabitable.